Last time, Nav1.8 claimed the first corner of the Grail. Today, we meet the quietest and strangest of the pain triangle, the youngest brother, Nav1.9. What makes it odd? 3 anomalies. 1. Like Nav1.8, it is tetrodotoxin-resistant, yet it is remarkably slow to activate, slow to inactivate, unable to fire a clean, crisp spike. 2. While other sodium channels wait for the voltage to climb before opening, Nav1.9 sits half-open near the resting potential, leaking a small, persistent current. 3. Because of this, it barely does any igniting at all. It does something else entirely. So what does it actually do? It sets the threshold. Picture a pain neuron as a machine gun. Nav1.7 amplifies the faint signal enough to pull the trigger. Nav1.8 fires the main burst of bullets. Nav1.9 is the finger quietly pressing on that trigger, making it easier to fire. Its persistent current nudges the neuron's resting state toward excitation, so a stimulus that shouldn't have qualified can still set off a train of firing. That is why it is most deeply tied to the lingering, ever-worsening ache of chronic inflammatory pain. Now the real mind-bender. The same Nav1.9 can leave you in agony or leave you feeling no pain at all. Its gene is called SCN11A. One class of gain-of-function mutation makes its current too strong, and patients suffer episodic, unbearable pain. But another gain-of-function mutation makes the current so strong that it pins the neuron in a depolarized state. The cell drives itself into inactivation and can no longer fire at all. And the result is congenital insensitivity to pain, feeling nothing even when injured. One channel over-tightened in 2 directions. One is fire, the other is dead silence. For all its importance, Nav1.9 is notoriously the hardest channel for patch clamp engineers to handle. Why? Transfected into the usual lab cells, and it often lingers inside the cell, refusing to traffic to the membrane, its current too small to record. So for a long time, people could only work on native dorsal root ganglion neurons, painstakingly isolating its slow current from a crowd of other channels. That is exactly why a truly stable Nav1.9 recording system is, in itself, proof of a lab's hard capability. But precisely because it governs slow pain, resists tetrodotoxin, and sits far from heart and brain, Nav1.9 is a tantalizing new target for inflammatory and neuropathic pain. No drug aimed specifically at it has been approved yet, and several groups worldwide are chasing it. And whoever wants to prove a Nav1.9 drug is selective enough cannot escape that same instrument, the patch clamp, comparing it against Nav1.7, Nav1.8, and the channels that must never be touched, drawing out the selectivity fingerprint one by one. To recap, Nav1.9, tetrodotoxin-resistant, remarkably slow, leaking current only near rest. It doesn't ignite, yet it quietly sets your threshold for slow pain, and it hides the double paradox of causing pain and abolishing it. With that, the pain-iron triangle, Nav1.7 the amplifier, Nav1.8 the main carrier, Nav1.9 the threshold setter, we have now met them all. But a new question arises. These channels, buried deep in the body and so hard to record, how on earth do scientists coax them into a dish and under the patch clamp? Next time, we tell the story of the magic that turns ordinary human cells into pain neurons, induced pluripotent stem cells. Follow Channel Observatory, and we'll take them one channel at a time.